we were at NAM during our ARS training. And without taking much time, because we would like to hear from Dr. Umesh Reddy. And just in 2-3 words, today, this lecture is very prominent and very important for all of us, particularly for our students and scientists, because this translational genomics for improvement of horticulture crops, as we are working on horticultural crops, and all the modern cultivars are domesticated from the wild relatives that are still found and possess several important genes that can resist, tolerate, and avoid extreme biotic and abiotic stress. In the process of domestication, ancient farmers developed land races from the wild populations that are currently part of germplasm resources for several horticulture crops. So genomic research involving these resources allows us to understand extent of natural variation for various important traits and genetic bottlenecks associated with domestication and modern plant breeding efforts. These are the important points that Dr. Umesh Reddy garu will be, sharing with all of us today. And with these few words, I once again welcome our chief guest, Dr. Umesh K. Reddy garu, and also all my seniors, my teachers, my colleagues, my scientists, and particularly Dr. Ravichandran from NARM, and all my other colleagues. Thank you very much.
Dr. Rao, can you make it shareable?
Yeah.
No, you should allow me to share.
Yes, sir. Yes, sir. Okay.
Open this.
Sir, we allowed, sir?
Yes, yes, I can open. Can you all see my presentation?
Yes, sir.
Yeah.
Before starting this presentation, I would like to profusely thank Dr. Janaki Ram, who is my best friend and also Vice Chancellor, a very honorable and respectable Vice Chancellor of this great university. And I am very pleased to speak here because it is named after one of my favorite politician, Dr. Y.S. Reddy. Moreover, and also I would like to thank Dr. Dilip Babu and Dr. Rao for facilitating this talk. Today is Thanksgiving break in the United States. It is just like our Sankranti or Diwali. It is a great festival around. I just attended a party and came here. And one more thing I would like to say about Dr. Janaki Ram is, though it is off the topic, he is one of the best administrators. When I visited IRI several years before, with limited funding, he made that university so glorified. I mean, that department, horticulture department. With his existing limited funding, he made that place so beautiful. He keeps his surroundings always beautiful. That is the most basic character of any horticulture department. You know, the horticulture department should be one of the beautiful departments in the whole agriculture university. And that's what he really takes care of it. And I see in the newspaper and from friends, I always listen that there is not much funding going on there. But I'm sure Dr. Janaki Ram will make that place so beautiful and presentable to anybody, to any international visitor. And the introductory speech he gave is so relevant that identifying genes from the wild species and transferring into cultivated is very important, especially because we have left out several disease-resistant and genes for stress, abiotic and biotic stress genes in the wild plants during the domestication. So in fact, my research is totally centered around especially we work in the horticulture crops. Main thing is to understand how we can transmit these genes that are left out in the wild species, putting back into the cultivated species. So there are many students here. I would like to take this 50 minutes from here. So the talk is around 11:43 currently here. So my topic is translational genomics. So just before going into the genomics, we all know the DNA is very important molecule, and this is wrapped around nucleosomes. Nucleosomes and DNA collectively is chromatin. Okay. So 2 nanometers DNA, when it is wrapped around these nucleosomes, it becomes a fiber. And this fiber coils into coiled coils and eventually a 1500 nanometers chromosome. So that is how it is stored. So before genes need to express, it has to undergo a lot of changes. One thing is, since it is so wrapped around these nucleosomes, these beads made out of histones, this needs to be released by these chromatin remodeling factors. We'll bind it and distort this so that the important binding sites are released. And then the genes, especially the histone tails of these histone proteins, they need to get acetylated and demethylated before the genes express. So once the genes are expressed, that is called the process called transcription. Once transcription releases the messenger RNA, the messenger RNA is transported into cytoplasm, where it is translated into proteins. Okay? So that is all the whole process around which we will be working. So what is genomics? Genomics starts with next-gen sequencing that identifies a lot of genes, lot of sequences, from which we analyze and identify what are the heritable genes that are that can cause biochemical modifications. Transcriptomics is doing RNA sequencing in different ways. So that is all transcriptomics. This is all to understand the phenotype. Especially we work with the horticulture crops, melons, pomegranate, watermelon, peppers, chili peppers. And then proteomics, sequencing the proteins, and metabolomics is characterizing the metabolomes. Everything is towards integrating everything. Genomics, transcriptomics, proteomics, metabolomics is to get integrated to understand the phenotype. So we have been struggling for last several decades. That's during the post next-gen sequencing era. The resolution was so less. I mean, pre-next-gen sequencing era. The cost was very high. You see this green one, the cost is very high. The map saturation, the genetic maps we make are very low resolution. QTLs we identify are very low resolution. The markers are very less, the substantial time it takes. So almost it takes 4 or 5 years to characterize one single gene previously. But nowadays, post-genomic era, the cost is less. You see this green, the difference, the cost went down so much that if you just have money sitting anywhere, you can sequence and characterize the genomes. And the map saturation has increased. The QTL resolution has increased. Markers that we can use has a broad choice currently. So you guys, students, are very lucky to be born as students in this era because you have all these technologies ready-made for you. After completing your BS, MS, PhD in the university, when you go to the United States or any other developed nations, all these tools you can use. You don't have to worry that currently you don't have it. It is very important to learn basics currently, but one day you are going to use all these tools. And then you have all these technologies currently available that we never had this. So the sky is the limit. By the time you grow up, you might have achieved so many great things. So one important thing that is currently going on is genomic selection. That is the breeding material, germplasm resources. You can genotype and then identify the best candidates, best parents. Initially, Dr. Janaki Ram worked on bottle guard, and he identified best combiners, like having specific combiners, general combiners. But now you don't have to do that. Once you genotype, you can, and phenotype, you can identify the best combiners that have all favorable allele combinations. One time you need to do genotyping and phenotype, just one time. And then you can identify the best parents that have all favorable allele combinations and make crosses. And thereafter, you don't have to phenotype. You can just use those favorable allele specific markers to make your selections. So this will, great way it will reduce the breeding cycles. You can probably, combined with speed breeding, you can effectively shorten your breeding period for 2 years or so instead of a decade. So these kind of technologies are available and they are very easy to use. And then there are so many deleterious alleles, like the domesticated genomes, the plants, they have a lot of deleterious alleles causing them susceptible to various diseases and pests. So this CRISPR-Cas is a technology. You can design a guide RNA. Here is a bad gene. So the bad gene is converted into guide RNA. And this bad gene can be sent inside the genomes using agrobacterium transformation. Once it is in the genome, the CRISPR-Cas motifs that are associated with this guide RNA and this bad DNA, they will go and identify the endogenous genes. See, this green gene has homology here. So this bad gene is removed by making this double stranded breaks. So the bad genes can be broken into pieces. That is what this technology is. So today I would like to present some scenarios that we have worked in our laboratory, published in our laboratory, about the phenotypically discernible genes. Especially to start, we mainly work with watermelon, one of the important horticulture crop. I do not know about these days Indian parties. I have not attended for a long time, but wherever we visit any American party, there will be watermelon there for sure. Everybody either will start with the watermelon or end with the watermelon. If you start with watermelon, you will eat other foods less. That way you can maintain your body weight, right? So people like to eat some watermelon to start with. Okay, so watermelon is here. We should not call it as vulgaris. This is the old slide made in 2011. Now we can just call it a Citrulus lanatus, variety lanatus. We can call variety vulgaris, but it is a little bit old. Citrulus lanatus is good enough because earlier this Mucospermus was called as another variety of Citrulus lanatus. Citrulus lanatus variety Mucospermus, but now this is called Citrulus mucospermus. This is called Citrulus lanatus. So you can call it as vulgaris, which is nothing to worry, but Mucospermus has its own species status. And then here is another species previously called as Citrulus citroides, but now they call it Citrulus amarus. So Citrulus amarus, Mucospermus, you know, the mucus is around the seeds. That's why it is Mucospermus, and then Citrulus lanatus. So to start with, we have a cucurbit cap project. So all the cucurbit accessions in the country will be sequenced. So under this project, we are partners of that process. So we sequenced these 1,400 accessions of watermelon that currently available in our germplasm bank, and we generated 65,000 SNPs. And then these are 3 different cultivated groups, and these 3 are semi-domesticated groups like land races, and these are the wild species. So from this wild species, that is the Mucospermus, these 3 semi-domesticated or land races develop, from which, you know, concurrently this cultivated species develop. So we published this paper in Plant Biotech Journal, in which we have even published the whole genome sequence of watermelon. So in our lab, we work with insect resistance for the watermelon. The trichome length and the trichome density are 2 important features that resist insects. And also we have aromatic metabolite diversity also we study for this. So there are different kinds of trichomes in watermelon. These trichomes are the tiny hair-like appendages on the leaves and stems. And the glandular cell, at the end there is a glandular cell. And then at the end there is no glandular cell. So the glandular cell here has 8-celled glandular top, having a lot of aromatic and volatile substances that can provide insect resistance. So we did association mapping for this. So 170, the most diverse actions were taken. And then there is a motorized microscope using which we can count the trichomes through the softwares, publicly available softwares. And once the trichome density and trichome length are estimated, we can use appropriate models to associate the SNPs that we have identified. So here is the density, high dense watermelon types and low dense watermelon types, and long trichomes on some of these genotypes, and very short trichomes. So then we have found this is the called Manhattan plot. These are different chromosomes of watermelon. These dots are single nucleotide polymorphisms. So the association, in the sense, if any SNP is falling above this bar, that means that is associated with trait of our interest, that is the trichome length or density. So here is a streak of SNPs. So this kind of strong signals can be identified. So we have identified some genes. This is called tetratrichopeptide repeat. So there are tetratrichopeptide repeat genes that has another motif called threadoxin motif. So we have TTL3 and TTL4 are 2 different genes, but both of them are tetratrichopeptide class. So in Arabidopsis, we can characterize, because in watermelon, you don't have genomic resources. You can use Arabidopsis, where these are the normal trichomes in Arabidopsis. But the gene, if we silence, the way I told you how we can silence the genes, by sending a guide RNA and cutting these genes into pieces. So those genes are not there. If those genes are silenced, you see this good trichome will become malformed trichome, indicating that the gene we have identified is very important for the trichome. You see here, it's branched normal trichome, and it has become branchless and malformed trichome. So these malformed trichomes, these mutant lines, we can rescue them. So we can clone the gene, watermelon gene, and put it in the Arabidopsis. Then the function is rescued. Similarly, the Arabidopsis gene also can rescue it, and we can overexpress the gene to see if we can create strong and robust trichomes than the parents, than the original lines. And also we can do yeast-2 hybridization. So yeast-2 hybridization, many of you will not be knowing what it is. This is a protein called GAL4. People use this GAL4 protein for yeast-2 hybridization, which is very important for galactose metabolism. So if the TTL4 gene is attached to DNA binding domain, this gene is divided into two. This GAL4 gene is divided into two. One domain has DNA binding domain and another is called activation domain. So we can use this gene, our gene of interest, as a bait protein, and then any other gene we can test. And then from this test, what we can understand, the TTL gene, the gene of our interest, and the new gene that we want to test, if both of these two interacts, then there will be transcription. So these two are whether they will interact or not. If these two genes do not interact, that means they don't have any relationship. So this, using the yeast-2 hybridization, we have identified 74 interactions. So these interactions, we have seen what these are. One is TCP transcription factors, which are very important for the trichome development. And then these are another few genes that are previously published. And then they were known that they have actin, actin polymerization. They are very important. So these two genes are previously known to function for the trichome morphology. And then this is mutant, this is the wild type. And then here is the stain, toluidine blue. This toluidine blue could easily enter into the mutant. You see, because the cutin is kind of compromised. Cutin is damaged here in the QTL4 mutant. When we silenced this gene, the cutin also damaged. Because of cutin damage, the toluidine blue can enter and plant turns blue, whereas the wild type, that remains normal. Here you can see the grown-up leaf, the mutant and the wild type. The mutant, in the sense, just one gene is silenced, and that is causing this damage. So like that, we are characterizing currently some mechanisms for the insect resistance. And then citrulline is another metabolite. So we published a paper for identity, and in that paper, we have identified genes for the citrulline, in watermelon. I told you the watermelon scientific name is Citrulline lanatus. So citrulline is the Citrulus lanatus. Why it is Citrulus lanatus? Because it has this amino acid citrulline, which is very important, especially in the watermelon flesh and the watermelon rind. There is this citrulline. This can cure cardiovascular diseases. Recently one of the superstar in Karnataka collapsed with cardiovascular disease, cardio arrest. If you take watermelon regularly, the citrulline can protect you from that kind of cardiac arrest, especially the Andhra people. Karnataka, Andhra, Kerala people has a haplotype. In our heart, the myosin remodeling factor, one of it has a mutation. Especially if you are from the Vaishya community, you have a very strong haplotype of that. That's what recently a paper published. So you need to take citrulline to release your cardiovascular muscles. So this kind of genes we can use and develop watermelon varieties that have very high content of citrulline. You see, this is the natural variation for citrulline. Some of them are very high citrulline watermelon types already available in the nature. And also we worked seed-bound amino acids and total protein in watermelon seeds. You see, these days seedless watermelons are becoming fashionable. So what happens if we use continuously seedless watermelons? Eventually we will lose seeds. We will not have any seeds. But the seeds have these important features, like it has very unique amino acids that are not available in other seeds. And the total protein is also high. So with Texas A&M, we have collaborated and identified important watermelon types that have very high amino acids and high total proteins. And then we have identified some genes that are important for these amino acids and total protein accumulation in seeds. So these are all open source materials. I will share this PowerPoint with Dr. Rao. He can share with you so that you can read these papers. And also the watermelon currently used is seedless watermelon in the United States. 90% of, not 90%, 99% of people, they eat only seedless watermelon. So how can we get a seedless watermelon? By crossing diploid with tetraploid watermelon. A diploid is crossed with tetraploid, we will get the triploid progeny. If the triploid is pollinated, the fruits that are resulting will be seedless. So we identified the important genes, important mechanisms, especially the leaf is smaller in the diploid, in tetraploid it is larger. The flower is larger. It has a lot of trichomes, you see. The whole plant is bigger, but fruit is exactly same size. So some private companies that are producing tetraploid seed wanted to know why the fruit size is small and what can they do about it. Then we have identified some genes that are alternatively spliced. So this is genomic DNA, this is the messenger RNA. These are different exons and separated by introns. So in the tetraploid, we have identified, especially fruit-specific genes, this kind of splicing happening in the sense this exon is not there here. It is spliced out. And look at this one. This is very easy to understand. So in the diploid, you have full gene, but in tetraploid, it is a truncated gene, truncated expression. But this exon is very long exon. This whole intron is now behaving like exon in this tetraploid watermelon. So that kind of molecular mechanisms are important. Another student of my student, she did the research on understanding this tetraploid, diploid watermelons more into chromatin conformation and more aspects of transcriptional regulation. And that published in Plant Journal. Plant Journal used it as a cover article. Especially we use it to think in the nucleus, the chromosomes float around that do not have any importance previously, right? So we use it to think that the genes are located on a chromosome. They have nothing to do with the other chromosome, the genes that are located on the other chromosome. But in the nucleoplasm, when these chromosomes will swim around, they will swim different fashion when it is developmentally regulated, in the sense when the watermelon is growing. For example, some chromosomes, they will be inside, they will come out, they will come outside, or they will interact differently with the other chromosome. Or if you give any stress to watermelon, so the chromosomes will alter their, the way they are present in the nucleus. So there are more intricate DNA sequencing methods currently available to understand what is the pattern, how they will interact. So Like I said, DNA that is wrapped around this fiber and coils into coiled coils. And then this kind of chromosomes will have the compartments of this entangled DNA and protein structures. So these two compartments are called topologically associated domains. So one chromosome specific topologically associated domains will interact with the other chromosome. They will have AB compartments, each topologically associated domain. So they are altering into A, or B is altering into A, and A is altering into B. You see, this kind of science is totally new. That is coming up slowly. This is called epigenetics. So we understood, especially A means in the topologically associated domain that has more euchromatin, opened genes. And then B is silenced genes. So we have identified around 105 genes that are becoming active to inactive state, especially on chromosome 4. So the genes that are on the chromosome 4 are more vulnerable for silencing when you convert diploid into tetraploid. So that kind of molecular mechanisms we identify for the horticulture crops. We just don't grow crops. We do molecular genetics or genomics. And also we do grafting, especially watermelon can be grafted on the bottle gourd. And this paper is published in Plant Molecular Biology recently, last year. And this is very important. You see the bottle gourd and watermelon is grafted on that. And then what happens after grafting? So in the first season, we evaluated the graft. So this is the normal one, that is watermelon on top of watermelon. When the watermelon is a cyan, the fruit weight increase in both seasons, you see here. Whereas lacinaria is cyan, the fruit weight is reduced in lacinaria, that is the bottle gourd. So similarly, soluble solids did not change at all. Watermelon soluble solids are almost same. Statistically, no significance. But in the lacinaria, you see here, significantly soluble solids increased. Bottle gourd, when used as cyan on watermelon, its soluble solids increased. Its rind pressure in watermelon is increased. Rind pressure is very important for storage or transportation of watermelons, especially if you don't have cold storage. Watermelons will heat up and then blast, right? Break open. So having a good rind can protect them. So similarly, you can see here, rind thickness and rind pressure. Okay. So then what happens to the genes? We identified 422 genes moving from bottle gourd to watermelon or watermelon to bottle gourd. And here it is, bottle gourd is used as cyan. And here again, 441 genes are moving back and forth. So when we do grafting, it's not only just a support. In citrus, we do grafting. Lot of horticulture crops, we do grafting. So when we graft, the genes are moving from the rootstock to the cyan, and then from cyan to the rootstock, and then sometimes altering in subtle fashion the genomes, and also providing the resistance. So the fruit-specific, leaf-specific, stem-specific, root-specific genes, we found them moving back and forth across these grafts. And we confirmed it. So for example, here you see dark band here, that is in the bottle gourd band. When we grafted in the watermelon, you see there is no that band. It is, you can see clearly there is no band. But when watermelon used as cyan, from the lacinaria, it entered into the watermelon. You see that? You see that band there? That is coming in the watermelon from the lacinaria. So we could clearly prove that way. And then the most modern technology is QTL-seq to identify a bunch of genes. QTL-seq is all the traits are not manifested by major genes. They are manifested by small genes that are all over the genome. So those genes are called quantitative trait loci. So what plant breeders we have to do is we need to centralize or pool all these important QTLs or favorable alleles into pyramid into cultivars to make them resistant. Especially here, we made a magic population. Magic in the sense multi-advanced generation intercross. That is called MAGIC. Acronym for MAGIC is multi-parent advanced generation intercross population. And here are the Citrulus amorous types that I have shown you, the black-seeded, wild, white kind of watermelons that are wild, from which the watermelon is domesticated. So these are highly resistant to several diseases, gummy stem blight, powdery mildew, fusarium wilt. For all the watermelon diseases, they have resistance. So for 10 generations, they were intercrossed, and then 4 cycles of intercrossing of I1 generation. I0 generation, first lot of intercrossing was done. I1, again for I4, 4 cycles of intercrossing. And then from these segregants, without selection, we crossed with cultivars. All these important cultivars, watermelon cultivars, Crimson Sweet, Charleston Gray, all these are important watermelon cultivars in the United States. And then 4 cycles of intercrossing, and then seven cycles of self-pollination created this kind of recombinant inbred lines. Okay, so this kind of building mapping populations is a huge job. It requires a lot of discipline. Very experienced and talented plant breeders can take these materials to this stage. One PhD student created this population in Todd Wiener's lab. And then what we did in our lab, we made pools, that is 30 most resistant rails, 30 most susceptible rails we have pooled. So even after combined years of gummy stem blight rating also says whatever we have pooled is clearly resistant or clearly susceptible. Then we did genome sequencing. So how much time I have, Dr. Rao? I think I'm fine for another 15 minutes.
Yeah, no problem. Yeah, 15 minutes.
Yeah.
Definitely. Sure.
So we did a lot of sequencing. Those bulks. We have done deep sequencing to identify 7 million reads, 7 million SNPs, differentiating resistant bulk and susceptible bulk. So 7 million SNPs is a lot. So quite a bit. Exhaustively we have sequenced them and then compared them. So here is one software that helps us to analysis, especially the reference genome is amorous. So the 0 and below, especially if anything is crossing this blue line, you can say here is the resistant gene. So 7 million SNPs of resistant bulk and susceptible bulk are mapped to this reference genome, and then QTL-seq was performed. So that shows us like 3 important locations in the wild species that can create resistance. See, that have resistant genes here, and similarly one susceptible gene here in the Charleston Gray genome or the Lanatus genome. So this is how we can identify the QTLs of importance. So these QTLs can provide resistance to watermelon if we transfer them into cultivated species. So what those genes are, then especially one gene I would like to highlight and say is this one. So in the susceptible, all of them has the AG reads for the SNP, whereas resistance has A. So the additional G insertion caused susceptible. And then the resistance has 241 resistant reads, and then just 31 reads that are susceptible. So this is a very good gene. And then the gene name is machine synthase. So that's how the QTL-seq can be identified. And then we have identified these are the markers that can provide resistance, like the A gene is resistant here for this marker, A SNP, A nucleotide. Whereas if it has C, it becomes susceptible. You see, for all these are the important favorable alleles that are located on various chromosomes. So the little bit I will explain what is resistance and susceptible play, especially for the important genes. Coiled domain proteins are very important for resistance and susceptible. For example, the green is susceptible, and then the dark green here is the resistant. So here is a resistant wild material that has undergone these mutations and then provided moderately resistance. Because of these mutations, resistance is broken and became moderately resistant. And further, it has gone another more mutation and then produced a susceptible kind. You see, and again, new mutations accumulated, and then it became moderate, and again, mutations will happen to make it susceptible or resistant. So that's how these are the major populations of watermelon, because of this interplay of continuous mutations happening from one population to the other, and then through the intentional or unintentional introgression happening across these population groups, resulting into either completely resistant or susceptible or mixed populations. So that is a population genetic model to understand gummy stem blight resistance. And also we see these cultivars, whatever fruits we are eating, fruits and vegetables, they have a lot of microbes inside. You may think you are vegetarian. If you are eating a banana or orange or an apple or watermelon, not only you are eating vegetarian, but you are eating a lot of microbiomes. Some of the microbiomes are beneficial. Probiotic microbiomes are there in watermelon. And also there are some microbiomes that hasten ripening, because those things can enhance carbohydrate metabolism. And that's how they can participate in the ripening of the fruits. So that kind of studies we did. And also we worked a little bit on the sweet potato to understand, especially Dr. Raja Reddy's group in Mississippi State. Some of you may be knowing Dr. Raja Reddy. He has a very good phytotran, a $20 million facility, where he can, using a highly sophisticated computer technology, he can manipulate the carbon dioxide and water and temperature levels. So under that kind of elevated carbon dioxide and drought, we have studied the storage route development in sweet potato and identified some of these genes, microRNAs that are regulating some of the important genes to mitigate elevated carbon dioxide and drought stress. So that is one of the papers. And we work not only on watermelons, but we work on melons also, especially to understand the fruit firmness. We have identified genes. Some of the companies currently are pursuing genes that we have identified for fruit firmness in melons. If anybody of you are from Pulivendula, we have a place like Gandhi that used to have a lot of genetic diversity for melons, especially in that Papagni River. There is very nice melons called agrestis. So those agrestis melons are the founding populations of melons. I do not know where exactly is the melons domesticated in India, but melon is kind of arsenated in India. Melon has tremendous diversities in India. Melon in the sense muskmelon, you guys call muskmelon. We call it as cantaloupes or honeydews in the United States. So that is a multi-billion-dollar market, especially cantaloupes. And the fruit firmness is very important because fruits will become very soft and they are not edible then thereafter. So we have identified a very important genes for the fruit firmness. Especially, I urge Dr. Janaki Ram to send some geneticist or breeder to the area of Papagni River and collect these valuable agrestis melon types and create a kind of a project. We have these days USA International Development Funding. We can explore together to characterize those agrostis populations in the Papagni River of Gandhi. And then anthracnose is another devastating pest you all know in tomato. We have currently working with this. Here is a resistant tomato, and here is the susceptible tomato, where after 7 days post inoculation, you can see the infection. So we crossed this and made a recombinant inbred line population and identified several genomic locations that are contributing for anthracnose resistance. So these are the peaks, QTL peaks, under which we can identify genes for the anthracnose resistance. So we have identified some genes, and here I tell you what is happening. In the susceptible parent, that is in the cultivar, these genes are expressing really high, but in the resistance, they are not expressing. You see here, all green plant, red stage, in different stages, more or less, this gene is highly expressing in the cultivars, whereas this block of the samples are from the resistance. Similarly, this gene is also like that. In susceptible, there is a lot of expression naturally, whereas in the wild parent, there is less expression. So what is happening? Some of these genes, they are bad genes. They are not good genes. So they are hitchhiking the domestication. So they are kind of pasted with the domestication or domesticated genes. So plant breeders, when they are selecting for good tomatoes, they are natural. They are also selecting for these susceptible genes. Though they do not have any importance for the horticulture traits, they are just piggybacking, we call in America piggybacking or clubbing or being together. These genes are deleterious genes. So the genomes, currently the cultivated genomes, they are loaded with this kind of deleterious alleles. So we not only need to bring the new genes from the wild species, we can also remove the bad genes. That is what Dr. Janaki Ram's strategy. You don't have to have a lot of funding. With limited funding, you can beautify the department. You can make it very attractive. Similarly, the deleterious alleles can be purged out from the genomes to make them more resistant. So that is another project. And also we identify genes in the Arabidopsis for the aluminum toxicity and acidity, resistance to acidity, and then transfer this into horticulturally important vegetables. These heavy metals, here I have a message in the chat. Okay, yeah, yes, Papagni River is about 35 kilometers. Yeah. So the stress, heavy metal stress, we can identify using various approaches. We can identify important genes and move into these genes for plant breeding in watermelon or tomatoes or chili peppers. So that kind of study can be done. Especially the acidity is very important. This is all acidification. So the whole arable lands across the world is the pH is reduced, is getting reduced because of so many factors, right? Acidity. So it's a huge environmental constraint because the varieties that we have, horticulture varieties, especially even grapes or watermelons, mangoes, they are not suitable for growing in acidic soils. So we need to be looking how to mitigate this. So at very high acidic conditions, this nitrogen and phosphorus and potassium, their availability is reduced. Whereas aluminum toxicity or iron, copper, manganese, zinc, they are highly available under low pH. So in Arabidopsis, we explored natural variation. You see some of these varieties are naturally growing very well in low pH. And some of the varieties in low pH are substantially reduced. And some of them are moderately stressed. So we did a genomic study to identify two important genes, especially 2,926 genes are identified. From the stress experiment, especially I will show you one mutant, that is one bad gene that is silenced. So this is the wild type normal plant. So this is the mutant plant in which one gene is silenced. It is not showing any difference in control. You see here, in low pH condition, whereas the gene that do not have the CRK, the plant that do not have CRK gene, that is the mutant, is growing better than wild type. Wild type has CRK gene, and it is not doing well because of that. If the CRK gene is silenced, the plant is doing really well in low pH and in low pH and aluminum. You see how beautifully it is growing when this gene is knocked out. You can see clearly in control also this mutant is showing better than wild type. And in low pH, you see how superior it is comparatively to wild type. And in the low aluminum condition also how better it is. So similarly, the surface area, root length and root surface area, in both the mutant is showing better. So we are currently characterizing that gene, and we published a very important gene for arsenic. Arsenic is another important trait, stress. Arsenic can cause a lot of cancers and Parkinson, this kind of neurodegenerative diseases. It's highly poisonous with no beneficial effects of this hazardous material. So we have identified an important gene and published in very high impact journal. Many people are trying to transfer this gene even into rice. You see in the vegetables, especially the watermelons, if you grow in this kind of conditions, they accumulate all these heavy metals. Then we think we are eating fruits and vegetables, but they are actually poison because they have infested with heavy metals. And also we work with chili peppers. In our laboratory, we maintain 1,500 chili peppers, 1,500 different accessions. This is 1,000 pepper accessions were sequenced. These are the main groups. This is the baccatum peppers, annuum peppers, and Chinese peppers. And then especially the Chinese are very important in the United States. That is called habanero peppers. Habanero peppers have that distinct flavor and a very low pungency. See, that is generally people grill it along with meat. So these grilled peppers, especially called habanero peppers. So 250 Chinese peppers we have genotyped. And then with population genetic tools, we have identified that at one stage, these peppers divided into long peppers and then this blocky peppers. These are the habaneros, Scott bonnet, even ghost peppers are of this kind that have the fruit shape index of 1.16 something. whereas the long peppers have very high fruit shape index. So we identified genes that divided peppers into these two different classes, and then identified genes for the capsaicin, the pungency, because of this gene is non-synonymous HIP, that is changing glycine to aspartic acid is very important for accumulation of capsaicin. And here is the same gene, the anchor and repeat is accumulating capsaicin and fruit size. That's why you see the large peppers, they are sweet peppers, whereas short peppers, small peppers, Golconda type small peppers, they are highly pungent, right? So we are very careful when somebody keeps us a plate of small peppers, we don't eat them. But the big peppers, you can eat them just like salad peppers. So the same gene on chromosome 5 is in the similar orientation, just duplicated, one controlling capsaicin and another controlling fruit weight. So we are currently working more on those genes. And once the peppers are fed to the flies, they alter the gut microbiome in the flies. They increase probiotic microbiomes in the gut, in the stomachs of Drosophila melanogaster. So they have beneficial effects. And we have identified high vitamin C peppers and flavonoids, carotenoids, many nutritional components in peppers. We have done metabolome analysis. Currently, a student is working on all of them to identify the genes. Sorry, this is coming different place. This is the arsenic gene that we have published. I don't want to discuss much about it. Here is another important thing. The peppers, once fed to this Drosophila, in the male and control, the weight is reduced. Body weight in the Drosophila is reduced. And similarly, in the female, the weight reduction is much more in the female flies. Especially the triglyceride levels, you can see whosoever is having diabetic in this audience, you can see eating this kind of habanero peppers, the triglyceride level is reduced substantially in male flies as well as female flies. This is the total glucose levels. And then we did a lot of metabolome analysis to understand if you eat peppers, the genes that make vitamin D are threefold increase, upregulated. The vitamin E genes sixfold upregulated. And all these metabolites that produce various cancers are downregulated. So eating peppers can reduce the cholesterol. You can see here the control and the pepper fed. Here is dopamine. You feel happy eating peppers. Your dopamine level is increased. You know, that's why the capsaicin is kind of addiction. If there is a spicy food, we would like to eat more. You know, if there is no spice in chicken or mutton, probably we eat little bit, right? These peppers will make us to eat, but still they reduce the body weight. And we did even RNA sequencing, transcriptome, and identified some of the bad genes are downregulated and some of the good genes are upregulated. So that is very nice. And then some of the genes that are differentially regulated are listed here that have a lot of importance. So we are not only working with identifying the genes for the resistance, but also we have projects to help humans eating these vegetables like in watermelon, citrulline or capsaicin or various metabolites, how they can cure people from various diseases. Here is Alzheimer's genes. See, these Alzheimer's genes, some of them are upregulated, the bad genes are downregulated. So Alzheimer's in the sense, chadastam, the older people will have this disease, right? Alzheimer's, eventually you forget everything. And similarly, the Parkinson, cancers, cardiovascular diseases, obesity, we have identified so many genes that are differentially regulated once you fed peppers to the Drosophila. And all those Drosophila genes have human genes, exactly homologs to human genes. So we can relate whatever happens to Drosophila will happen the same thing to the human because we have the same genes, same metabolic pathways regulating us or predisposing us to diseases or to the various conditions. So we also work with pomegranate. So the whole aspects, I would say, my research, I can say what is the genetic basis for domestication and what are the fruit quality of or nutraceutical traits that are important in these vegetables and fruits, and what are the genes for the biotic and abiotic states. So this, some of my old students, all these grad students currently left. We have quite a diversity of people in my laboratory. And this semester, I am taking one student from Anantapuram, first time from my area. And, many students can apply and we provide the complete assistantship and full tuition waiver. What is required is you need to have a GRE score and a TOEFL score. And then some of students, especially, I forgot the professor's name. He contacted me and he proposed a student also. Currently, she applied. Probably the next semester we will consider her application from your university. Nakkalapudi Vanaja, some name, I forgot her name. I have her application. And then our funding comes from USDA, NIFA, and the Department of Defense, USDA ARS, NIH. This is another group of students. So thank you very much. If you have any questions, please do not hesitate. I do not know if I have taken more time than given to me. I apologize for that.
Thank you very much, Umesh. Really, it's a fantastic lecture loaded with so much of science covering different horticulture crops. And definitely we look forward to work with your laboratory and your university. Thank you very much, Dr. Umesh.
Thanks, Jankiram.
Sir, thank you very much, sir. And you have given a lot of information on genomics and genes responsible for biotic and abiotic stress and nutraceutical. So all excellent, sir. Any doubts from any audience you can raise?
Thanks, Rao Garu.
Sir, Umesh, I have a question.
Yeah, Ravi.
Umesh, actually this year, many of the universities have exempted from students taking GRE and TOEFL.
Yeah.
What about what is the status of your university?
It's not unfortunately exempted. Some of the good universities, there are more progressive universities than what I'm serving. They are proactive in doing that. Hopefully our people also will do it. Because our students, they cannot do GRE very well.
Yeah, that's what I'm saying.
That is the huge this thing. But eventually, well, it is possible.
Especially from agriculture background, if students can be exempted, it would be better. You will have more number of students from India.
Yeah, I think so too. I am currently director for graduate admissions. I am trying with administration if that is possible.
No, you tell me that what are the current areas of research in horticulture, so that our the production can be jumped up.
Just finishing.
What are the current areas
for
Indian conditions?
It's not only like what I have presented. We are molecular geneticists, but there are many horticulture departments. Horticulture is working with production technology, like the the production technology like agronomy or weed control or plant pathology, virologists. There are all kinds of groups working everywhere. So ours in India, horticulturists are very strong. And I'm sure great scientists. Today I'm facing great scientists developing a lot of varieties and technologies. The South Indian horticulture, is very strong. The vegetables that we have in South India, the way they taste, are nowhere in the whole world. First time when you come to the United States, first thing you notice is the vegetables are not as tastier as grown in South India. You see, it's great germplasm. And this is, as Dr. Janaki Ram said, this is second most university that is premier for horticulture alone, right? Right, Dr. Janaki Ram? That's what I said.
Yeah. Right.
Okay. Thank you, Umesh. Thank you.
I have one. First of all, sir, I had a very good lecture, sir. Excellent. Actually, the very complex subject, you have made it very simple. I think you are a very good teacher. First of all, I have to openly appreciate, sir. Second thing is only just basically I'm a plant pathologist. Why just please enlighten me why the mutants or variants will be more in viruses when compared to fungus and bacteria at the genomic level, sir, please.
Say it again, Raghuvar. I couldn't catch the last point.
Pardon?
Again,
I couldn't catch the last point. Can you repeat
okay,
the
sir. My is actually generally we are observing more mutations or more variants in virus particles when compared to fungus and bacteria in genomics generally. Why it is? That's why just I asked you just for that.
In fact, I do not know. Probably what you are mentioning. Recently, I attended the Cucurbit Cap meeting. The virologists were saying that a lot of new virus is coming all the time. And whereas even much more, I know you guys have a great virologist there, Sai Kiran. Yeah, I think in the university. I have really do not have clear answer for that, Raghuvar. Why virus is more variable comparatively to the other diseases.
Yeah, Dr. Gopal is virologist.
Dr. Gopal.
Dr. Gopal is our virologist.
Okay.
Yes, sir.
And he has some papers also, I think.
Oh, very nice.
With Namaste, sir. I am Dr. K. Gopal, working as a registrar here. And one more thing is, one of the associate professor working with you, Padma, she is my classmate in UC at the SPA College. Hello, sir.
Hello. I couldn't hear. Can you please?
Padmavati.
Yeah, Padma she is my wife also. We both together do research. And she used to say that, her classmates are deans and directors in your university.
Yes, sir. Yeah. So my colleague in classmate in the UC, sir.
Very nice. What is your name, sir?
K. Gopal.
J. Gopal. That's what she mentioned. Today, now it is almost midnight for us. So she is in bedroom. But I will tell that.
We have made morning for you. Sir, thank you. Really, sir, topic is very wide topic, and again covered all the crops, sir. And you're taking examples of fruits, vegetables, watermelon, tomato, chili, all really a great lecture, sir. And we have excellent students here who want to pursue molecular biology breeding aspects in our labs. Last year, actually, we tried. At that time, I think it was time was over for some admissions like that. I contacted.
Yes, we should take as many students from our from our universities. The only bottleneck is as soon as we say GRE, nobody will apply here.
Yes, sir. Yes, sir.
That is what currently happening. But once the GRE is cleared, you know,
Yes.
the students who come here can save a lot of money.
Yes, sir. Yes, sir. Yes.
You know, the assistantship is so good, and the tuition is, per semester we have $24,000 worth of support per student.
Yes, sir.
Dr.
So,
Umesh, we have
yes, John.
English language laboratory.
Very nice, very nice. I know Dr. Janaki Ram will do this kind of things.
Yeah. You know, encourage the students for GRE and TOEFL also. That is
exactly wonderful, wonderful, John.
So in future, definitely we'll also.
Yeah. You know, ladies and gentlemen, the Vice Chancellor, our Honorable Vice Chancellor can not only speak English, but he knows German also. Sometimes he will talk in German with us. So he can come up with this kind of ideas to make students proficient in English. It's not a big deal. Yeah. You see, I'm from ZPH school. I can speak English. Yeah.
Sir, any queries from anybody? Otherwise, we will go for vote of thanks by Dr. R.V.S. Kiriti Garu, Director of Research, Dr. YSR Horticulture University.
Good morning to all. I take it as a privilege to propose vote of thanks for this webinar that was conducted. And at the outset, I would like to thank our Honorable Vice Chancellor, Dr. D. Janaki Ram Garu, who came out with a novel initiative for the faculty with the Global Connect series to get exposure with regard to the modern breeding techniques in crop improvement. Thank you very much, sir, for creating this opportunity and roping in Dr. Umesh Reddy Garu, an eminent scientist. Dr. Umesh Reddy Garu, Professor of Biology, West Virginia State University, USA, an eminent scientist at international level in modern breeding techniques, who has delivered a lecture today on translational genomics for improvement of horticultural crops. He dealt an array of horticultural crops, especially with regard to watermelon and the physiological attributes that are associated with the resistance, especially the trichomes, and how they have transformed into the trichome chemistry involving the particular techniques. And also at this level, I got the privilege of going through his research paper on chili diversity, which was shared by our Honorable Vice Chancellor. And definitely, sir, our faculty got exposure to modern breeding techniques, molecular mechanisms, genomics of grafting with regard to biotic and abiotic stress resistance, QTL, sequence analysis for gummy stem blight, fruit ripening, drought resistance in tomato, especially with elevated carbon dioxide concentration. And he was also dealing with muskmelon diversity, especially in the Papagni River Basin, where the Siddhavatam area is very popular as far as the genetic diversity is there. Our university scientists are also involved in collecting the genetic material, sir, for your information. And he also dealt with tomato anthracnose resistance, heavy metal, especially with regard to aluminum and arsenic compounds resistance. And what else? The gene silencing. On the whole lecture was dealt with the genetic basis for domestication, resistance, breeding, as well as fruit quality in horticultural crops. Thank you very much, sir. We are really honored to have you, sir, today as our first speaker in this series, which was the initiative taken by our Honorable Vice Chancellor. I hope all our faculty got benefited from your lecture, sir. And we are expecting many more lectures from you, sir, even in this series or otherwise for any a program that is being conducted by our university, sir, because really it is very informative. And I also thank all our university officers who has shared this, we have attended this webinar, and all the faculty, students, and the research station scientists, everybody who have participated, including our zonal research heads, research station heads, and all the faculty that is involved. Thank you very much. And I thank the organizers, especially our director, industrial and international programs, who has taken pains in conducting this particular webinar, first of its series. with regard to this Global Connect series. And I thank him for giving this opportunity to all our faculty members. Thank you, one and all. Thank you very much.
Thank you. Thank you.
Thank you, sir.
very much.
Thank you, Janakram. Thank you, Umesh. Thank you.
Thank you very much, Umesh. I'm really extremely happy to see you.
Thank you very much, sir.
Sir, thank you, sir.
Thank you, Dr. Janakram. Thank you, sir.
My regards to Padmagar.
Sure, thanks. Yeah. Again, thank you very much.
Thank you. And,
for organizing this great lecture.
And
especially Dr. Rao and Dilip Babu.
Yeah, we will also do something with Papagani, germplasm. So
that's nice. That's very
shortly
good.
We are
that
going
can
there.
do like world focus, international focus.
Yeah,
Dr. Umesh Gar, I'm here. Thank you very much for inviting me.
Great. Thank you, sir.
Your lecture has been an eye-opener to all of us. It's wonderful. We have got a very strong group at Lam working on this.
All right, thank you.
And especially on the areas you talk about, bioenergetics is wonderful.
Dr. Dilip Babu was first to connect with Dr. Umesh.
Yeah, he is.
And I have disturbed you in the late night also. I'm sorry, but
well, it is pleasure. Thank you. Take care. Yeah.
Thank you very much.
Thank you. Good night. Good day for all of you.
Thank you.
About 2.8 crores released, sir.
Thank you, sir.
Thank you, sir.
Thank you. Thank you.
Thank you, sir.
Thank you.